This is our lecture introducing amino acids and going into ketones and ketone bodies and things like that, other things that can come from proteins and amino acids. So just the basics as a review for amino acid structure. We have kind of a set of levels that we can apply to thinking about amino acid structure or protein structure, starting with what kind of amino acids you have in the protein, what order are they in. Then after you get through with that, which is the primary structure, then you can start thinking about, okay, are they going to bind into a helix kind of shape, like similar to the way DNA binds, or are they going to be more in the pleated sheet version, which would be something like if we just took it and had a more flat group of amino acids or a flat bonding of amino acids and fold it like a piece of paper. That's why they call it pleated sheet. It's not perfectly folded here, but you get the idea. So it's something like that, and then we could have interactions with the different folds. So that's the secondary structure. The tertiary structure would be where you have those interactions with the different folds. So if we have our pleated sheet amino acid, maybe this amino acid here is going to bind to this one here. Maybe it's more like that. So all sorts of different ways that those amino acids can bind to each other, particularly because a lot of them are going to have different side chains as part of the primary structure or as part of the secondary structure, and those side chains are going to interact also. Now, once you have this really folded, whether it's a pleated sheet or a helix, once it's really, really folded, or a combination, once it's really, really folded, then we have what's called the peptide. So this would be just an individual small protein structure. Now we can take each individual peptide and have a bunch of them and bind them together to form a quaternary structure. So here they have 4 shown, but it could be 2, it could be thousands of different peptides all bound together. So again, we're thinking of binding not just the primary structure, but also side chains, and not in just a 2-dimensional form, but in a 3-dimensional form. So that's just the basic general structure of how proteins are set up, how they're made in the body. When you're making them in a cell, you start by stringing the amino acids together, and then they begin to form the shapes and the bonds and more bonds to get the full protein structure. Now, when we are thinking about breaking down our amino acids, that's what catabolism means. So, when you're building something, that's anabolism. When you're breaking something down to get energy or its component parts, that's catabolism. So when you're catabolizing your amino acids, you can be doing that to extract their amino acids that are going into the formation of the entire protein. Once you have those amino acids extracted or broken out of the main protein structure, you can oxidize them for ATP. So you can be taking electrons away from them and using them directly to make ATP. This would be an example of substrate level oxidation rather than what we've been doing with the electron transport chain and all that with our chemiosmotic phosphorylation and that sort of ATP production. This would be a much more simple, basic form of ATP production with just directly oxidizing the substrate. But in order to do that, you have to first convert it to whatever amino acid it is. You have to get it to acetyl-CoA. So there's various mechanisms for that depending on which amino acid we're talking about. Other amino acids are called glucogenic amino acids. Glucogenic, it means that they are able to create glucose. So you can convert the glucogenic amino acids directly into glucose. Now, if we're talking about skeletal muscle, we can take those glucogenic amino acids and turn them into something called alanine, which is another amino acid which can then get out into the blood from the skeletal muscle cells over to the liver, and the liver can use alanine for gluconeogenesis, which is, we're going to talk about that, the glucose and the alanine relationship in the liver and with the skeletal muscles in a little bit. Now, other amino acids are considered ketogenic amino acids, and those can be converted into keto acids, also known as ketone bodies or ketones. We've mentioned these before. Briefly, they are something that can be used by cells for energy. Again, we have to get them to Succinyl Coa, but we're going to be able to run them through the Krebs cycle and do our chemiosmotic phosphorylation with the electron transport chain and all that kind of stuff once we do get them into the form of Succinyl Coa. Now, ketones or keto acids, keto bodies, these are special because they are something that can be used by the brain cells. So remember, in your brain you can use glucose as a substrate for energy. You can use ketones, keto acids for energy. And we also did learn that you can use lactic acid as energy. That's sort of the new knowledge there. It was thought before it was only glucose and ketones, but now we know that also lactic acid can be used for energy by the brain. So anyway, I'm digressing. The whole point here that these keto acids or ketones are very important because they're a good source of energy for the brain when your blood sugar is low. So we're going to talk a bit about that later on in this lecture too. Now we can also just kind of be recycling our proteins or the amino acids from the proteins, upcycling them, I guess, if you want to think of it like that, because you can convert them into other things. You can convert them into triacylglycerol. You can create glycogen. If you do, gluconeogenesis, then you can do glycogenesis too. So there's all kinds of different things you can do. You could just make new proteins if you have need of different proteins than the ones you have. So you can always just break apart these proteins and get those amino acids out and use them for other things as well. Okay, so in the fed state, so remember the fed state is when you have eaten, you have absorbed nutrients, so you have glucose in the blood and you've got plenty of energy available. There's a excess of energy, so you don't need to be breaking down your fats or your proteins for energy. And really, ideally, it's not great to use proteins for energy in any case because it's just more of a valuable molecule. If you think about when humans are evolving and what nutrients are readily available, protein would be one of the more difficult ones to get enough of. So it's important that you don't use proteins too much for energy. Also because they have lots of other uses. You can make enzymes with them, you can make hormones with them, some neurotransmitters, all kinds of things, structural components of your cell. Lots of things are made using protein. Now that said, we are going to use some protein for energy at different times, but just not in the fed state. There's no need for that. Now if you're in the starvation state, and remember, this doesn't have to be a long starvation state. Like, you don't have to be literally starving. It just means that, it's been 6 hours or so since your last meal. So overnight, this happens, unless you eat right before bed and you only sleep for a short amount of time, and you're going to enter that starvation state. So what happens there is you actually lose a bit of muscle protein. So if you think of the contractile filaments that muscle is made of, the actin and the myosin, those are made of proteins, and those are what makes the muscle strong and able to contract well. The more you have, the more you can contract. So you actually lose some of those during those fasting periods or the starved state periods. And why you're doing that is to keep the body full of energetic supply. So if you're short on glucose, which you would be, you would have low blood sugar if you haven't eaten for a while. You need to have your ketones. You need to have, that is a source of energy. We know that your brain can also use lactic acid that the astrocytes were making when they break down glycogen, but that's just going to be dealing with your brain, right? So you also need to have the ketones to help supplement that source of energy. Okay, so when we break down proteins, it's called proteolysis. So lysis just means the destruction of or the breakdown. So proteolysis is breaking down the proteins. And when you're breaking it down in muscles, we are just really thinking about releasing proteins from muscles into the blood. So if you think of those protein structures that you've created in your muscles and almost like a reserve of energy or amino acids that you can release when needed. Now, the thing about proteolysis in muscles is that what they release is not really representative of what they contain, if that makes any sense. So the proportions of what they release, they are releasing mostly alanine and glutamine. These are the, two main amino acids that are released from the muscles. They account for about 30% of the total amino acids that are released from your muscles during a starvation period. But alanine and glutamine don't make up 30% of the amino acids in a muscle during any other time. So we are simply breaking down proteins and creating alanine and glutamine through a series of steps in order to release them. Now, why those two are the most important ones, or why those are the ones that are being released in kind of a greater quantity than what they represent during a non-starvation state, is that alanine can be used by the liver. We mentioned this before. The liver can use that alanine to make glucose. So there's a cycle for that of chemical reactions. Pretty basic set of chemical reactions. Glutamine is actually tied up in that alanine cycle as well, but it's also used by the kidneys for creating glucose. And your intestinal cells, whether we're talking about the smooth muscle or the columnar epithelium in the intestines, they can use those for energy as well. Okay, so some amino acids, 3 of them, are called branched chain amino acids. These branched chain amino acids are abbreviated as BCAA, branched chain amino acid, and these are where we get our alanine and glutamine from in our skeletal muscles. So when we are breaking down proteins within the muscle, we're getting out these branched chain amino acids and using them to construct alanine and glutamine. So just remember that alanine and glutamine are coming from branched chain amino acids. Now what branched chain amino acid is, you can read that, it's a little fuzzy. I know it just doesn't zoom so very well. But what this says is that they have branched chain or branched aliphatic side chains. Aliphatic means as opposed to aromatic, like an aromatic is got the ring structures. These don't have that. Their side chains are just chains of different structures with carbons and hydrogens on them. Now the 3 of them, the 3 branched chain amino acids, leucine, isoleucine, and valine. So these are our 3 branched chain amino acids. When we're breaking them down, so we're breaking down our branched chain amino acids by branched chain amino acid, let me write this down, amino transferase in our skeletal muscle. It's written over here, but I'm thinking you probably can't see that. if we're talking about the liver, it's broken down by a branched chain amino acid, alpha keto acid dehydrogenase complex. So that's happening in the liver. Why we're using that in the liver as opposed to just the plain old amino transferase is that the liver doesn't have the branched chain amino acid amino transferase. So it has to use this. And there are various things that would activate this or deactivate this, just like we saw with the pyruvate dehydrogenase complex. It's an enzyme similar in that way. Okay, so how do we oxidize our branched chain amino acids? So as we are looking at, being in the starvation state, and we are looking at releasing alanine and glutamine. So remember, these are the two that we are, looking for to release from the muscle, and they are made by branched chain amino acids. So that's where we get our branched chain amino acids. So this could be leucine, isoleucine, or valine. Okay, so it could be any one of them. And we'll just follow that. So let's not actually start here. Let's start in the muscle. Okay, so we have our branched chain amino acid, and if we're in a fed state, we can be doing protein synthesis. If we are in the starvation state, we can be getting energy simply by oxidizing, so substrate level oxidation. Remember, we mentioned before that we can just directly oxidize our amino acids and get energy, ATP, straight out of that just by a basic substrate level oxidation. But as we're doing that, we also have this happening. So when we're looking at, sorry, not glutamine, I'm getting my tongue tied, not glutamine, glutamate is GLU. So remember glutamate, we've seen glutamate before with aspartate in the malate aspartate cycle. We've seen glutamate just in a few different forms, few different ways of, being an intermediary kind of product, and that's essentially what it's going to be doing here. We also see alpha ketoglutarate. This should sound familiar to us too from that malate aspartate cycle. I shouldn't try to write and talk at the same time. It doesn't work. Alpha ketoglutarate. And remember, the difference between these two is simply an amino acid, whether we're aminating or deaminating the molecule. So if we're aminating it, or we're taking the amino acid away from it, we're going back and forth from those two molecules. So this is a very simple thing to do. Just take an amino acid away, add an amino acid, and you have this little cycle here. But in the process of this, a couple of different things can happen. One, the glutamate can easily be converted into glutamine. That's what the GLN is for. And that is what is released by the skeletal muscle, and that's going to here a piece of smooth muscle in the intestine, which can just use it for ATP production. Over here, though, we have a little bit more going on. So if we kind of zoom in, we see alanine being also created as a side product off of this little circle here. In this case, we are using pyruvate in order to feed into the circle and to get the alanine, as opposed to going over this way to get the glutamine. And the alanine is also released and goes to the liver. Now we're going to get into the alanine to glucose cycle, but we essentially go from alanine through pyruvate and then taking the pyruvate to get a glucose. It really isn't much more complicated than that. But what's interesting here is that once we get the glucose, so the liver has made glucose now, in this case from alanine, and that glucose is now in the blood, but if this glucose came from somewhere else, it would be, just now you're no longer in the starvation state, right? You have glucose in the blood, your blood sugar is up, you no longer need to be breaking down your proteins and getting glutamine and alanine because you have glucose now in the blood. So if we follow this little chart here, let's switch colors so we can follow the glucose. Here comes the glucose into the cell. Glucose getting converted into, through glycolysis into pyruvate, and that would, of course, give us energy, through the Krebs cycle and all that. But it's just feeding into what's going on back here as well with our glutamine production. So going into between glutamate and alpha ketoglutarate, and with an enzyme that can deaminate these things, we can use our pyruvate to feed into the cycle of creating more glutamine. So when we go into a starvation cycle again, we can reverse the process and get more glucose back out of it from the liver. Okay, so here is the glucose-alanine cycle in a bit more depth. Trying to get that to show up nicer. There we go. Oh no. There. Okay, so over here at this picture, we have the muscle cell. So this would be skeletal muscle. There's the blood, so that's just the intermediary. That's where, things will travel through to get to the liver. So hepatocytes would be over here. So this is literally just zoomed in on what we saw before and kind of focusing a bit more on the relationship between glucose and pyruvate, glutamate, glutamine, alanine, and things like that. Actually, we're not looking at the glutamine. We know we could get glutamine out of this, but we're focusing on the alanine because we're dealing with the liver rather than the intestine. Anyway, so let's start with our glucose. Wherever this glucose came from, in this case, it's coming from the hepatocytes, coming from the liver, but wherever that glucose might have come from, maybe, like if it was from being in the fed state, what I'm trying to say is the source of the glucose doesn't so much matter as long as we have the glucose at the start of the process. Now, we know as we are in a muscle cell and we're going to do glycolysis, we know that we're going to be adding hydrogens to NADH while we do glycolysis. We know all of that. We're going to get 2 ATP out of that glycolysis and 2 pyruvates that could be going into the Krebs cycle. If we had, if we were using energy, if we were in the fed state, if that glucose were coming from the fed state, that would just be going to get energy. But since we are looking at how we deal with being in the starvation state, we're not going to be sending those pyruvates into the Krebs cycle. Instead, what we're going to be doing is breaking down our muscle proteins. So we're going to be doing proteolysis here. That gives us our amino acids. NH4, that's just, the byproduct that's part of the amino acid. That's, what's going to be the group that's moving around, as we aminate and deaminate would be the NH4. And then we get glutamate, okay, in this case. So remember, we had glutamate for the production of glutamine and also for alanine. Here we're looking at alanine. It could just as easily be, going for glutamine too. I'll put that over there if we were going to the intestines. But here we have our glutamate interacting with our alpha ketoglutarate and the pyruvate as well. So as these three things come together, what needs to happen is the amino acid has to go from one thing to the other. Remember, we said that is the only real difference between glutamate and alpha ketoglutarate is an amino acid moving from one molecule to another. So here we have an amino transferase. In this case, it's an alanine amino transferase, so very specific for alanine. So you know, if we were doing glutamine, it would be a different amino transferase, but here it's alanine, so we're going to use the alanine amino transferase, get alanine out of this process. That alanine goes into the blood. Here it is in the liver, in the hepatocyte, and then we're just going to reverse the process. It's really just the exact process in reverse, using this alanine amino transferase again to just, you know, transfer that amino acid now, but instead of, you know, going from pyruvate, glutamate into alanine, now we're going from alanine into glutamate and pyruvate, so the opposite way. Alpha ketoglutarate is there to accept the amino acid from the alanine and the amino transferase. Now, two things are going to happen here. We've already seen how, you know, we can take pyruvate and we can do gluconeogenesis to create glucose. It takes energy, so we got to use energy for that. It's going to be really just backwards glycolysis if you think about it, because instead of making 2 NADH like we did in glycolysis, we're kind of taking the hydrogen off of NADH and getting NAD plus instead, and we end up with our final product of glucose. Now we also have some waste products being formed here as we look at our glutamate. The glutamate can do a bunch of different things, so we're going to see some more things that glutamate can do within the cell now that it's just here. You know, we've seen the malate aspartate cycle, we've seen it as part of other metabolic maps, but we're going to see a couple more before we're done with glutamate with the semester. But then we're going to have these waste products as we go up here. Urea. This is something that is not a desirable thing. It's just a, you know, something that needs to be gotten rid of. And is excreted with urine. And we also have a urea cycle to deal with the urea, which we'll do in a few weeks as well. Okay, so that is the glucose-alanine cycle in the liver. Now ALT, that is the amino, or I can't say that for some reason, the alanine amino transferase. ALT is a liver enzyme, obviously. It's an enzyme that is measured when you're doing your blood work. If you have high levels of ALT in your blood, it can often mean that your muscles are being broken down at too high a rate. So there's a normal rate of ALT that you should be having. You know, typically when you get your blood drawn, you are in the fasting state, so you could have higher levels of ALT, meaning that you're doing more proteolysis, you're creating more alanine, so you're making more ALT, alanine aminotransferase, in order to, you know, convert that alanine into glucose. So that would be a within normal limits kind of situation. It's when it gets too high and remains too high for a longer period of time that doctors will start to wonder what's wrong with it. Could it be, you know, muscle wasting? Could it be a liver disease sort of situation? Okay, so another metabolic map here. Here we see so here's a branched chain amino acid. Let's just write that down there so we all see it. Sometimes when they have all these letters on here, it gets hard to remember what the letters are standing for. So remember, this could be leucine, valine, isoleucine, any one of the branched chain amino acids. Now, remember we also mentioned that when we're dealing with branched chain amino acids, there are specific amino transferases for them. So a branched chain amino transferase, so that would be the BCAT right here. So I'll just write that, branched chain amino transferases. And there would be over here as well. Now we have this thing here, the BCKA. This is a branched chain keto acid. Okay, so this is something that we can get from the branched chain amino acid. And remember, a keto acid we can use for the Krebs cycle, we can use for energy. So this is something that is a valuable thing that we need to get when we need energy. And then there's one last thing, abbreviation here, which we need to mention, and that is the branched chain alpha keto acid dehydrogenase complex. So that's a big mouthful. Branched chain alpha keto acid dehydrogenase complex. Okay, so you see the dehydrogenase, you know that's an enzyme, and a complex just means it's a kind of complicated enzyme. It's an enzyme that has different components. Branched chain, you know that it must be coming from a branched chain amino acid, and it's going to be a keto acid because of the K in there, so a branched chain keto acid dehydrogenase complex. So the only thing we have left to wonder about is this alpha part. We haven't gotten to that. It just has to do with the structure, the chemical structure of the amino acid, or of the, keto acid, where the double oxygen bond is, if it's, on what carbon it is, if it's, on the same side, if it's, right next to each other or spread out. There's a slide that has pictures of it, so we'll talk about that in a bit. But that's what it's dealing with. So here, because we're thinking of keto acids, we are thinking of making energy. We are thinking of the Krebs cycle. Here they have it abbreviated as the citric acid cycle, but this is just the Krebs cycle. Every time you see this circle. And here it is over here again. Over here they just put some details onto it. So alpha ketoglutarate, malate, oxaloacetate. We know all those details of the Krebs cycle. But what we have going on here is, just to start, our branched chain amino acid. And we can go in, it's just showing that it can go in multiple directions here. Okay, so we can start with, let's actually start over here with our branched chain amino acid here. There is our branched chain amino transferase to, transfer the amino acid from one to another. So here's our alpha ketoglutarate again. There's our glutamate. There's glutamine. We're making glutamine again. So that's that whole process that we've seen. While we're doing that, making this glutamine, here we are with a branched chain keto acid now. So what are we going to do with that keto acid? We're going to work on it with the branched chain alpha keto acid dehydrogenase complex. So that's the enzyme that works on the branched chain keto acid. And we're going to use that enzyme complex to make acetyl CoA, and that can go into the Krebs cycle. Energy can happen, as it does from the Krebs cycle. We can also be taking that Krebs cycle and branching off at an intermediary product and creating pyruvate again, right? And having that turn into lactic acid. That's a possibility, and back and forth. So we know that that can happen if we're doing the Krebs cycle. So we're either going to be, getting energy out of this, or making pyruvate. If we were in the liver, say, we could be doing gluconeogenesis, like that, various things. But same over here. They just, I don't know why they put it over here again with slightly less detail, but there's just the branched chain keto acid, and there's that that complex, that enzyme complex that can get acetyl CoA and start it into the Krebs cycle. But what they're just showing here, why they had it in two. Anyway, I'm not sure why they put the Krebs cycle on their choice, but the important part here is that the glutamine and the glutamate, these can be recycled, and the alpha ketoglutarate. So as the glutamine can go backwards, we don't just have to do alpha ketoglutarate into glutamate into glutamine. We can do the backwards way, glutamine into glutamate, creating in the end alpha ketoglutarate, and we could do protein synthesis here. So we can make the branched chain amino acid in this way using the same enzyme. So it's still just the branched chain amino transferase, but it's doing something different here. Okay, so this slide looks very complex, but it's not, because we're not sitting here memorizing all of these different parts. What we're just looking at here are the three branched chain amino acids. These are our three that we mentioned before: leucine, valine, and isoleucine. And we're just looking, really, the whole point of this is whether they are ketogenic, glucogenic, or both. So some can be both. A lot of amino acids are both. So all amino acids are going to be able to be broken into something. And remember, when we're ketogenic, we're going to be making ketoacids. If we're glucogenic, we're making, pyruvate, glucose, things like that. And then if we're both, we can do both. So leucine is ketogenic. Its specific keto acid that it makes is something called HMG Coa. So HMG Coa and leucine go together. You get HMG Coa out of leucine, and from HMG Coa you can convert that into acetoacetate, which is a specific kind of keto body or ketone, and then acetyl-CoA. So acetyl-CoA can go into the Krebs cycle. Keto bodies, are important for, obviously, the brain, things like that. Okay, now for valine, valine is glucogenic. So what it's going to do is end up creating pyruvate and glucose. It does it through an intermediary product called Succinyl Coa. Succinyl Coa, that comes, it's used in the Krebs cycle. It's where we branch off, or close to where we branch off, to go back into creating pyruvate and doing gluconeogenesis. So that molecule itself, Succinyl Coa, is how we get to, or link, our amino acid to pyruvate and glucose. Also, acetyl-CoA. So you see, anytime you're going to use acetyl-CoA, or see acetyl-CoA for the Krebs cycle, then energy, right? Now, isoleucine does both. You can get keto bodies and glucose/pyruvate out of it. So when you get a keto body, it's an alpha keto body, which we're going to look at soon, the difference between an alpha, beta, or a gamma keto body. But in this case with isoleucine, it's an alpha keto body, which gets turned into acetoacetate, just like over here with leucine, same thing. And acetyl-CoA for the Krebs cycle, but also we could turn it into pyruvate and glucose via Succinyl Coa, just like we did with valine. So the purpose of all these pathways is really just to see, here's leucine, there's the end product, acetoacetate, and acetyl-CoA. Here's isoleucine, that's ketogenic and glucogenic, so we're going to get acetyl-CoA, and then where is our acetoacetate? Over here to acetoacetate. And then for valine, we're getting into Succinyl Coa, which will go up to acetyl-CoA. So it's just showing the end products. You don't have to be able to memorize this, obviously. Okay, now this is showing the Krebs cycle. But it's not showing it from an oxidative point of view. It's showing it from a ketogenic versus glucogenic versus both amino acid sort of situation, and where they go into the cycle, or where they feed into the cycle, each specific amino acid. Again, this is not for memorization. This is for simply seeing and being able to notice where these amino acids go into the actual Krebs cycle. So on this chart, there is something that is a bit unclear before we get into it, so I just want to point that out. That where it says ketogenic amino acids, which we'll zoom in and we'll look at these more closely, but where it says that they are ketogenic amino acids, almost all of these are also glucogenic. There's only a couple that are not also glucogenic. So leucine, which we saw as one of our branched chain amino acids. Oops, that's in the wrong spot. Leucine. And lysine. Those two are strictly ketogenic amino acids. The rest of these in here are also glucogenic. Now, over here on the glucogenic amino acids, they just left valine out for some reason. So anyway, I put that on there. That was another one of our branched chain amino acids, along with leucine and isoleucine. So we just added that on there. Okay, so let's look at this little chart here. See if we can get this to be focused. Okay, so it's really just a depiction of glycolysis, right? And we know glycolysis can go backwards into gluconeogenesis. So we can either start with glucose and go into glycolysis and then the Krebs cycle, or we can be doing it backwards and doing gluconeogenesis. So let's start from the beginning. Let's go forward. So we start with glucose. We're doing glycolysis, breaking it down. We're getting our pyruvates here, and then yielding eventually acetyl-CoA that can enter into the Krebs cycle. Now, before we even leave our pyruvate though, here come our glucogenic amino acids. So alanine, we saw that coming out of the skeletal muscle in a starvation state. Alanine was that one that the skeletal muscle released and that the liver cells, the hepatocytes, could use for gluconeogenesis. Cysteine, glycine, serine, threonine, and valine, which is one of our branched chain amino acids, they can go into pyruvate and then up into gluconeogenesis, okay, and make glucose, so glucogenic. Now, once we have pyruvate and we get acetyl-CoA, because they can also do that, right? So once you get pyruvate, you can make acetyl-CoA as long as you have pyruvate dehydrogenase and go into the Krebs cycle. So now we're at acetyl-CoA, but we could also have keto bodies coming from our ketogenic amino acids forming into acetyl-CoA. So these ketogenic amino acids include phenylalanine, tyrosine, leucine, which we've seen, lysine, tryptophan. I think everybody knows what tryptophan is. You've heard of that one. That's the one that helps you make melatonin. And then isoleucine, or other branched chain amino acid, going into acetyl-CoA. Okay, so we have our keto bodies, ketones, and we have acetyl-CoA going into the Krebs cycle. Okay, now once we're in the Krebs cycle, we start turning the Krebs cycle, and here at alpha-ketoglutarate, this is another entrance point for other amino acids. Arginine, histidine, glutamine, so we saw glutamine again being released from skeletal muscle. This would be for like smooth muscle in the intestines, you know, using glutamine for energy. You put one of these guys in here and run the Krebs cycle and you're getting energy just like you would from any other entrance point into the Krebs cycle. Also proline and glutamic acid enter there. Succinyl Coa. So we know from our previous slide that isoleucine was one and valine was another that went into succinyl Coa. So here it is in the Krebs cycle. Methionine is also one of those that we have not seen before that enters in at succinyl Coa. But we can go right from succinyl Coa to pyruvate for gluconeogenesis. Okay, then finishing up the Krebs cycle, we just have two more steps to make. At the fumarate part of the Krebs cycle, phenylalanine and tyrosine can enter into the Krebs cycle, at the very end, at the oxaloacetate part, aspartic acid and asparagine can enter into the Krebs cycle there. So this would be where the keto bodies from our ketogenic amino acids end up, forming or creating, our ketogenic amino acids, I should say, make the keto bodies plus the acetyl-CoA, which can go into the Krebs cycle. Up here at the pyruvate is where the glucogenic amino acids can enter into the Krebs cycle, or creating glucose going backwards through gluconeogenesis, or via Succinyl Coa through gluconeogenesis. Okay. Oops. Page is stuck. Okay, so the three main types of keto body are acetoacetate. So we saw that from our two glucogenic, our two ketogenic amino acids, beta-hydroxybutyrate and acetone. Only two of those can be used by the brain. That would be the forms of acetoacetate and beta-hydroxybutyrate. So here's what they look like generally. Just note our oxygen double bond. Acetone has one of those. Acetoacetic acid has two, plus a hydroxy group. And then beta-hydroxybutyric acid has one plus two hydroxy groups. So these are just the three main kinds of keto bodies. And then we can get more deeply into specific keto bodies, like the one we saw coming from like leucine, things like that, and whether they are alpha, beta, or gamma, depending on where these little oxygen double bonds are, what carbon they're on, are they on the same side of the molecule or on opposite sides of the molecule. So for our description of whether a keto acid is an alpha keto acid, a beta keto acid, or a gamma keto acid, we are thinking about what number carbon that double oxygen bond is on, and also if it's on the same side of the molecule. Remember, if you think back to chemistry class, if we have, that's not like a good picture, it's the boat conformation versus, why am I unable to draw this right now? Chair conformation. I'm doing a terrible chair conformation. Anyway, here's the main part of the molecule. Here our constituent parts are on the same side, so that would be cis. Cis means same. Here is our main part of the molecule, and here we have our constituent parts on opposite sides. So that would be trans, like across from each other. So that's what we're looking at here, whether it's cis or trans. So if we look at an alpha keto acid here, we've got a trans situation, but those carbons are right next to each other, or the oxygen double bond, I should say, are our own carbons that are right next to each other. For the beta keto acid, we have here a cis conformation, but notice that there's a carbon in between. So we have the oxygen double bonds spaced by one carbon. There's a separation here. And then for the gamma keto acid, just find the oxygen double bonds, and here we have a space of two carbons, and this would be a trans conformation because they're on opposite sides here. So examples of alpha keto acids are pyruvic acid, so our pyruvate that we have seen all the time, that is a type of alpha keto acid. Alpha ketoglutarate, that's another one that we've seen a million times. HMG CoA, that's one we just met from leucine. Alpha keto isovalerate, that's one we have not seen. But all of these are just examples. There are just many, many examples of every kind of these keto acid, depending on what amino acid they came from and what has happened to them. Okay, so looking again at our HMG CoA, So HMG-CoA, that's an alpha-keto acid like we just saw. We know that it comes from leucine, which is a branched chain amino acid. So how are we going to make that? So ketogenesis is the production of keto bodies, or ketones. Okay. And what we're looking here, so this whole big blah 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 right there, this is just the extended version of HMG-CoA. Okay, that's just the actual word for HMG-CoA. We're just going to call it HMG-CoA. Now here, this pink thing, HMG-CoA lyase, this is just the enzyme for HMG-CoA. Okay, let's see, here's another enzyme, dehydrogenase. Okay, so we're just dealing with two enzymes and a couple of intermediary products. So we start out with our HMG-CoA. We act on it with our HMG-CoA lyase, so right there. And we end up with, oops, there's acetyl-CoA that can go to the Krebs cycle. And here's acetoacetate. Remember, we saw acetoacetate as one of our main keto bodies. Okay, so this is the carbon skeleton of the ketogenic amino acid. So in our case, it was leucine because we're dealing with HMG-CoA, but it could also be another ketogenic amino acid. Any of the ones that give us acetoacetate as our product. Now, we're going to use the dehydrogenase on the acetoacetate, and we can end up with two specific keto bodies, our acetone and our our beta hydroxybutyrate, which is over here. So we got these two keto bodies out of that. Okay, so not really difficult steps, just two enzymes, two things. We're able to get energy out of here and the keto bodies right there. So that is how we made our ketones out of our ketogenic amino acid. Okay, now ketones can come also from fatty acids, and they often do. So in this diagram, this is showing fat. So it's showing how we get ketones from fat. We've watched ketones coming out of leucine. We just saw HMG-CoA coming out of leucine, so out of a branched chain amino acid. But here we can also see it coming from a fat. So it would be the same product in the end, and the same, once we have the HMG-CoA, it would be a different form or a different way of getting it out of the fat. But once you have this process would be the same. All of this would be the same once you have that particular substance. So no matter where we're getting our ketones from, here when they say a fat depot, they're meaning a fat storage, so adipose, say, or ingestion. The liver also produces fatty acids, puts them together, conversion, all that kind of stuff. And here are some various, keto bodies that we can be making. We've seen, HMG-CoA. Here's our acetoacetate. That was back here. Let me find it. Lost it. Oh, right here. So here's acetoacetate, right? So that intermediary product between HMG and two ketones. Okay, so when they say beta oxidation has to do with lipid breakdown, so we know that, and breaking it down to get these particular keto bodies. Now, once we get acetyl-CoA out of them, so we can go right into the Krebs cycle, which is right up here, you know, just like we did with glucose, glycogenesis, or glycolysis, going down from glucose into pyruvic acid into acetyl-CoA. Here, our acetyl-CoA is just coming from keto bodies. Same way that we saw in the previous slides, and we go to the into the Krebs cycle. Now, the last point I want to make very briefly, I know this video is going on kind of long, I'm losing my voice because it's gone on for too long, but I just want to point out the difference between ketosis, which is having ketones in the blood, this is how you cope or how you deal with being in the starvation state. So this is a source of energy for your brain when you don't have glucose handy. This is the whole point of what we've been talking about this whole time, is to have ketosis. So this is a good thing. This is something that you want. Something not good is to have too much ketones in your blood, too many keto acids, and that would be ketoacidosis. This is where the keto acids are being kind of your only source of energy. So you're in the starvation state for too long, so you're breaking down your structural proteins. At too high of a rate, so you're losing muscle mass, you're losing cellular structure, your enzymes might not be working so well, you might be getting deficient in some proteins, but a more immediate danger is that keto acids, by nature, they are acidic, and they can mess up the pH of your blood and cause you to be more acidic. So if you've ever heard of, somebody being in ketoacidosis, which you probably have, such as, somebody being a diabetic and going into a diabetic coma when ketoacidosis becomes too severe. That is because when a person is severely diabetic and they're insulin resistant, or they don't have enough insulin, which means that their cells do not have access to glucose for ready energy, they can end up relying too much on ketones. It's as though they're in the starvation state. They could have plenty of glucose in the blood, but it's not able to get into the cells. So that becomes a starvation state, essentially, for their cells. And they start, breaking down proteins too much, too often, for too long, and you end up with ketoacidosis as a result. So that's obviously not a good thing. But that ends our lecture on ketones and proteins and on glucogenic amino acids in our next video.